Thank you. I'd like to thank the committee for selecting my abstract. This is a collaborative effort between my lab and Adele O'Toole's lab in Queen Mary and Kevin Mills' mass spectroscopy lab in UCL. And the problem on the left-hand side of this slide is that a major risk factor in eczema is the reduction of expression of filaggrin, either through mutation or just through a reduction in expression. And this inhibits that function that's attributed to filaggrin of keratin filament collapse, and therefore we get barrier disruption. But we wanted to ask the question, are there other mechanisms that lead to barrier disruption in eczema that may or may not be exploitable? So on the right-hand side here, we can see our sort of very sort of small experimental protocol. We have a look at models of eczema by knocking down filaggrin by shRNA, and we interrogate these by phosphoproteomics and by RNA-seq, and then we correlate our findings to eczema samples from the Bangladeshi community in East London as part of the Tower Hamlets eczema assessment. And with these, we have a valuable resource where we can analyze nearly 1,000 tape strip proteomes and do immunofluorescence on a more limited set. And then we put these data sets together to try and determine new mechanisms of barrier disruption that could be druggable in eczema. And to cut a very long story short, we did a lot of phosphoproteomics on our knockdown cells, and we identified our top 10 down-regulated kinases, and 4 of these were MAP kinase related in these boxes here. On the right-hand side is our the results of looking for BMP and TGF-beta related gene expression in our shRNA knockdown keratinocytes, and we could see that there were increases and decreases in expression of BMP and TGF-beta signaling. Oh, that's wrong. Let's try again. There we go. So we looked at our eczema samples to see if what we were seeing in our models reflected the real patients. And you can see in the top panel there, we can see an increase in fluorescence of ID1, which is a well-known BMP SMAD158 target gene. And we see it's increased in filaggrin heterozygotes and compound heterozygotes. And we could show this downstream activation of BMP signaling by immunofluorescence. And by Western blots in tape strips, we could see that our compound heterozygotes had a significantly increased amount of phosphorylated SMAD. Phosphorylated SMAD is inversely correlated with filaggrin overall and positively correlated with keratin too. So it suggested there may be some kind of rescue afforded by having increased BMP signaling in these eczema samples. And to test this further, we treated keratinocytes with recombinant BMP6, and we could see a dose-dependent increase in the expression of filaggrin monomer in human keratinocytes. And this was concomitant with an increase in keratin-1. Conversely, if we treated with a BMP receptor inhibitor called DMH1, we could see a dose-dependent decrease in filaggrin expression, processing, and keratin-1 expression. The other thing we saw, if you remember, was the decrease in ERK MAP kinase signaling, and we could confirm this in our shRNA knockdowns. We had 3 different biological replicates, 3 different knockdowns, and we could see a reduction in phospho-ERK1/2, and we could see that reflected in panel B in our eczema samples. Wild types with high levels of filaggrin had high levels of phospho-ERK1/2, and we could see as the genotype becomes heterozygote to compound heterozygote, we get a reduction in phospho-ERK1/2. And we could see a correlation between filaggrin and phospho-ERK in our samples. We treated panel C as a treatment with BMP of normal human keratinocytes, and we can see that BMP treatment was activating one of these MAP kinases, P38. So we also looked at the global differences in phosphoproteins in our phosphoproteomics of our shRNA knockdowns, and only one group of proteins was significantly downregulated throughout all of our phosphoproteins, and that was very interestingly desmosomal proteins, so this clamp that holds keratinocytes together in the epidermis. And we could see a large number of these genes indicated by the proteins, rather, indicated by the blue circles here, where these phosphoproteins were downregulated. And what was interesting is when we looked at our RNA-seq data, we could see that actually changes in desmosomal protein expression, desmosomal gene expression, was quite modest. However, by Western blot, we could see downregulation of various components of the desmosome in our Filaggrin knockdown keratinocytes. Finally, a student in the lab, Kosuma Raman, who visited for 6 weeks and managed to do all of this bottom half of this slide, showed that 2 components of the desmosome, desmoplakin 1 and 2 and desmocollin 3, were mislocalized in our Filaggrin knockdown keratinocytes. So we can see here cell surface staining in our desmoplakin stain and our desmocollin 3 stain, which is lost in our Filaggrin knockdown keratinocytes. So how do we put all this together? And this is my sort of conclusion slide. We found by proteomics, rather, of the tape strip samples that we had in abundance, thanks to the Tower-Hamlet's eczema assessment, we could show that a subset of these had high levels of phospho-SMAD158 staining, or 158 activity. And this subset of high SMAD158 patients also had high levels of, or higher levels of, a number of key desmosomal components. So where we're currently at now is looking to see if there are biomarkers of this increase in desmosomes, and whether identifying other proteins that correlate with BMP signaling, i.e. high levels of SMAD158 phosphorylation, comprise genes involved in barrier rescue. And this network here are the 20-plus proteins that we saw correlating to high SMAD158 levels. And in red here are our key proteins that were in kinases and BMP6 and their potential interactions with these proteins. And we propose that these small subset of proteins could comprise potential therapeutic targets that could activate the BMP signaling pathway in eczema to rescue desmosomal function. So I'll leave it there, and I'm happy to take any questions.
Thank you. Okay, any question? So as I understood, these are possible treatment targets to improve the dysfunction of the epidermis, right? So you...
this is very early stage, but we'd like to think that increasing BMP signaling would enhance desmosomal function and therefore rescue barrier function in a subset of eczema cases. We'll need to do much larger studies.
Of course.
This is a pilot study of only 20 samples, but we have access to nearly 1,000, so we can really look at these biomarkers in more detail and try and nail down the ones that correlate with BMP signaling and may constitute potential therapeutic targets, or at least point to mechanistic targets.
Perfect. Thank you. Thank you. Thank you so much.